Well, I'm Steve Rappaport. I was involved with the committee when we had the first two Exposome workshops. I'll give a brief history and see all this in context. So David asked me to address the question of what is the exposome. It's like beauty in a way. I have my views of it, and I'm sure you have yours as well. I think the main goal of the exposome, its greatest utility is going to be in the area of disease etiology, finding causes of disease. Well, everybody dies of something. What do we die of in the world? Well, 2/3 of us die of chronic diseases. Infectious diseases are on the wane, even in the poorest countries. And it's people dying of mainly cardiovascular disease and cancer. People are getting older. As they live longer, they're more likely to get these diseases of old age. What causes these diseases? Chirag alluded to it briefly. Is it the genes or the environment? The reason I got interested in the exposome initially was this discovery that the genes actually had a very small role in chronic diseases. I was shocked. My parents had horrible cardiovascular health, and I thought for sure that I'd be dead by the time I was 60. But it turns out that's not the case. And we can weigh these genetic versus exposure-related risks with a term called a population attributable fraction. And for those of you that aren't epidemiologists, you probably haven't heard of this. So we abbreviate it PAF. And it's just basically, it represents the proportion of the population that, or the proportion of the disease cases that would be prevented if the causal factor were eliminated. So if we eliminated the gene, or if we eliminated an exposure, it would be the proportion of lives that were saved or diseases that were prevented. So I recently combined two databases to get some idea as to the proportions of cancer that we could attribute to the genes and then the non-genes, the exposures. And so this slide shows all of the deaths in 2010 worldwide. There were 50 million deaths, and of those there were some 7 million cancers divided up into 25, 26 categories. And I was able to get population attributable fractions for most of these cancers from the Swedish Families database. In a wonderful article, it was compiled by Kari Haminki and his colleague Zen, I can't remember her first name, published in 2002. And so by combining these things, you can see in this slide the total deaths in 2010. Say for lung cancer here, it's about 1.6 million deaths. And the red blotch at the end represents the deaths that would be attributed to genetic factors, heritable genetic factors. And you can see for all of these cancers, the red blotches are pretty small. And overall, it is 3.8% of all cancers can be attributed to the genes. Some have more. Breast and prostate are in the 10 to 20% range, but these are the hormones play a big influence in these cancers. And hormones, as we'll see, are genetically controlled. So those are exposures that we have to consider as part of the exposome as well. So it's an interesting idea. But the main point of this is to say that if we're really going to find causes of disease, we have to stop looking so hard at the genes, because it's not going to do us that much good. And these genetic PAFs were calculated in a way that make them contaminated in a sense, because they also include the shared exposures. And remember that these PAFs were determined from families, and families share a lot of environmental factors. So in fact, the PAFs are probably smaller than what they seem to be. And this fits other epidemiological evidence. It's known that genetically stable populations have different cancer rates over time. Cancer rates increase and decrease, even though the genetic population doesn't change really. When people from one genetic pool move to another country, they migrate, you see that the cancer incidence after a generation starts to take on the characteristics of the new country. So these are, again, it's empirical evidence supporting this idea that the genes are not really the major drivers of cancer. What about exposures? Well, we don't have nearly as high technology generating exposure data, whereas we have GWAS and whole genome sequencing that we can use for understanding genetic factors, and Chirag alluded to that. For exposures, we still ask people what they're exposed to, and that's the nature of the technology. And here, based on very simple questionnaires, really, you can see that about 1/3 of all cancers in the world have been attributed to a relatively small number of risk factors, smoking being the biggest one. You saw that lung cancer was the most prevalent cancer in the world, 1.6 million deaths, almost entirely attributed to smoking. People didn't smoke, there would hardly be any lung cancer. And there are other things too: diet and exercise, alcohol, unsafe sex, indoor smoke, and so on. So the point is that we can attribute more than 1/3 of all cancers to this very crude collection of exposures that we've been able to compile over the years from questionnaire data and personal interviews. So if we want to find the unknown causes of cancer, we can continue to elaborate genetic factors employing even higher tech omics if we want to. And now the goal is to go to genome sequencing for everything. And that can be done, and it'll probably be done relatively inexpensively, but it has explained relatively little of the risk. So we're not really going to get too far along that line. If we want to elaborate the exposures, we still rely on these low-tech solutions. But this has explained 1/3 of the cancer risk. So we need to level the playing field. We need to get better measures of exposure, and the purpose of this workshop is to really look at one avenue that will potentially provide much better evidence of exposure. Well, this, of course, this dichotomy between high-tech omics being able to tell us something about genetic influences on disease and low-tech exposure assessment being able to give us limited information about exposures motivated Chris Wilde to write his provocative editorial back in 2005. And he really saw this exposome as a complement to the genome in studies of disease etiology. He saw this as a tool, this combination of tools that we'd be able to use to really find the causes of disease. This paper was published in 2005. I read it in 2009. I didn't see it when it first came out. But it just so happens I had just been appointed to this committee for the National Academy to develop workshops for new science and technology that had implications for environmental health. So I came to my first meeting. Some of the people in the room were here. We were all suggesting possible topics, and I said, well, why don't we have one on the exposome? And people looked at me like, exposome. So that's like the totality of all expos. I mean, what is this exposome? So we debated that for a while, and then once the idea got out and I told them about Wilde's really cool editorial, and we read it, we decided, yes, it might be worth a try. So we put on this workshop. It was a very provocative meeting. I know that several of you were here. It's one of the most stimulating meetings that I've ever attended. And it got me so energized that I went back and I immediately started writing a perspective piece for Science. Now, I never published anything in Science before, and I was very dubious as to whether they would go for something like this. But I wrote it, I called them up. They said, well, sounds weird, but we'll take a look at it. And to make a long story short, they did publish it. And that really heightened, I think, the visibility of this whole idea. And it motivated the National Academy to put on a second workshop to deal more with the practicalities of implementing methods to measure the exposome, and considering that you have both internal and external sources of exposure, as we've heard. And this, of course, has really heightened the awareness of the exposome. This slide simply shows the number of papers or times the word exposome was cited as a keyword in a published paper. And you can see from Wild's commentary, very few citations until we had the first National Academy workshop and the science perspective piece, and the second National Academy workshops. That really set an acceleration in the understanding and awareness of this concept. And then a lot of other things have happened since. The EU really picked up on this and put major funding into studies in Europe to start elaborating the exposome. There was this Phenome Center that was established at Imperial College London. Then last year, or 2 years ago, NIHS set up its first exposome-focused NIHS center, which is called Hercules, and it's at Emory University. And then just this year, David mentioned that they're taking these funds from CHEER, or from the children's study, to set up this big network of laboratories to do high-tech children's exposure assessment. So how does the exposome actually get measured? I think one way to get at this idea is to think of the pathways that cause chronic disease. So this is based upon an earlier model that was published by Eric Schott, and he saw the genome and the environment as contributing to disease, but he saw them as essentially influencing the transcriptome, and then the transcriptome would go on to lead to the disease processes. And when I looked at that, I said, well, G and E have to be important, but if E is going to communicate with the body, it has to do so through chemicals. The chemicals in the body are either the proteome or the metabolome. So E really has to influence those two sets of chemicals if it's going to contribute to disease traits. So the other part of Schott's model recognized that as the disease process begins, it alters the transcriptome. This is the so-called effects of the disease. So the transcriptome changes, it's going to change the proteome and the metabolome. And Schott recognized that you would get these secondary traits or comorbidities. So this model really tells us that the etiology of disease is complicated because we have this causal pathway and we have this reactive pathway. And we have to take them both into account if we're going to actually find out what causes disease or what is the result of the disease. And it essentially all happens through chemical communication, because everything in life is communicated chemically. There's nothing that we can do or think that isn't translated into a chemical in one way or other. And these chemicals communicate with other, and that's what life is all about. And chemistry is the language of life. So there are all these G-related factors. These are chemicals that are derived from the genome. We know a lot about the DNA, the RNA, and then the protein. The proteome contains all of these really functional proteins, enzymes, cytokines, receptors, transcription factors. We have small molecules that are signaling molecules, neurotransmitters, hormones, lipids. All of these arrive from the genome. Okay? So a big part of the metabolome is really molecules that are motivated by the genome. And they probably are responsible for contributing this genetic contribution to chronic disease risk. But we also have these, what I call E factors. So these are the factors that are either exogenous to the body or are in the body but have sort of exogenous elements. So we can think of antigens, foreign DNA and RNA, pollutants, of course, and then the microbial metabolites, very important group of molecules that are intimately involved in all areas of our lives. Drugs, and then, of course, foods, nutrients, and toxins. So these are coming in largely from outside, or in the case of the microbial metabolites, from inside. And these molecules alter communication, and the beginning of disease is really related to these changes in communication of molecules. So we know we have genetic modifications that take place when small electrophilic molecules that are part of the metabolome interact with the genome. We call these mutations. These small electrophilic molecules also interfere with functional proteins. We call these post-translational modifications. So we get damaged DNA, we get damaged proteins as a result of interactions with these small molecules. We also get a whole host of epigenetic modifications that are caused by both small molecules and proteins, transcription factors. So it's very complicated, and you can see that you have both the causal pathway and the reactive pathway that are very much involved in all of this communication. So how are we going to capture all of these exposures? So this is a figure that was published in our science paper. And the main point of the figure is to demonstrate that you have these external sources of exposure, and we have these internal sources of exposure, including what we called at the time the gut flora. Now we call it the microbiota, all the genes as the microbiome. But to me, what's this saying is that exposures are chemicals. And the blood exposome includes all chemicals in the body. So if we focus on the blood, it gives us an entity that's going to integrate all of these exposures from all of these external and internal sources. And the microbiota are among the most important of these factors. We have 25,000 human genes, and yet in our bodies, there are 500,000 more genes derived from the microbiota. So 90% of all the protein-coding genes in our bodies are microbial. It's an enormous number, and those protein-coding genes are there to produce proteins that metabolize small molecules coming from our diets, and those molecules are transmitted back into the body. They're intimately involved with establishing the human immune system in early life. They set up axes with all major tissue groups. So it's enormous. I wanted to get a glimpse of the exposome. So a couple of years ago, I worked with people at IARC when I was on sabbatical, and with David Wishart, who's here. And I collected all of the molecules that were easily accessible and had been measured in significant numbers of human beings. So we knew the concentrations of these chemicals, and we could also classify these chemicals into 4 broad categories: drugs, foods, pollutants, and endogenous. And these are the cumulative distributions of the concentrations of those chemicals that have been reported in human blood. You can see that the range, the dynamic range of blood concentrations is enormous. It's 11 orders of magnitude. It goes from femtomolar to millimolar concentrations. You also see that 3 of the 4 categories are highly overlapping. Each category itself has about a 7 order of magnitude difference in concentrations. And 3 of them are essentially indistinguishable. These are the foods, the drugs, and the endogenously derived molecules. And this says that food-derived molecules, endogenous-derived molecules, are in the same dynamic range as pharmacologically active agents. What are these things? These are some from foods. I'll quickly go through these because I'm about out of time. But each of these is very biologically active. Some of them have been associated with disease, both as protective agents like sulforaphane, which protects DNA. TMAO has recently been recognized as a potentially major source of cardiovascular disease. Hormones, we know, are important molecules. The sex hormones, stress hormones, malondialdehyde results from lipid peroxidations, very reactive molecule, probably carcinogenic. Homocysteine, involved with cardiovascular disease. And cholesterol, the number 1 studied exposure. Some drugs, aspirin, very protective. Digoxin, extremely toxic. And here's the environmental chemicals, some metals, DDE from pesticide exposure, the perfluorinated compounds, benzene, which I studied for years, codeine from smoking, and so on. The main thing that comes from this is that there's a 1,000-fold difference between the environmental chemicals and everything else. And that creates real problems, measurement problems. It's hard to measure things down in the noise. So when you do untargeted analysis, there's real issues related to measuring environmental chemicals. One of my colleagues, Dinesh Birapal, was doing this chemical mapping with a program he wrote called Cytoscape. And this is a map. It's a chemical space of those 5,000 or those 1,500 molecules. And the size of the dot represents the blood concentration. And the dots that cluster with each other usually have particular chemical properties that are similar, like all the lipids are here, the sugars are over here, steroids. Here the PCBs are this little porcupine-looking structure. The metals are all up here, and so on. And what this shows is that there's extraordinary diversity, chemical diversity, in the blood exposome. There are more than 100 classes of molecules, which, again, brings detection issues to bear. For each of these molecules, we looked at the number of PubMed citations that were available, and 336 of them had been associated with chronic diseases, either cancer, cardiovascular disease, or respiratory disease. And the size of the dots here represent the number of citations. The most highly cited exposure is cholesterol, 4,500 citations. But you can see we have citations for all classes of chemicals, for environmental chemicals, for food-derived chemicals, drugs, and endogenous-derived chemicals. And that says that epidemiologists look for sources of disease with chemicals from any sort. They don't care where they come from. They just want to know that they cause disease. And this also says that these small circulating molecules that could be mushed together as the metabolome provide one very important avenue for characterizing biologically relevant exposure. It's not the only one. It's not only small molecules. There are proteins that are important. There's foreign DNA and RNA that we could evaluate with metagenomics. These are important too. So to get at the whole exposome, it's not just the metabolome, but the metabolome is a very important piece, and it's very accessible now. So I'm almost done. To put this all together, one would do an exposome-wide association study, which is generically similar to a genome-wide association study. So we would apply EWAS to biospecimens from healthy and diseased subjects, find those features that are different, and then follow up those features to determine where they come from, whether they're really causal, and so on. And so it's a two-stage process, a discovery phase followed by large population studies to understand the true etiology of these factors. And going back to the disease model, it tells us a lot about which biospecimens are going to be the most useful for EWAS, because we have this causal set of biomarkers and we have this reactive set of biomarkers. And if we want to find causes of disease, we want to focus here. If we're interested in early diagnosis of disease, we'd want to look here. But in any case, if we're interested in disease etiology, causes, these reactive biomarkers are really going to obscure the causal pathways. And so validation of exposure biomarkers is going to require getting specimens that were collected well prior to diagnosis, what they call prospective cohort studies. That's the only way you're really going to find the causal factors. So this is the untargeted EWAS design. We start with the blood exposome. We have diseased and healthy subjects. We do untargeted analysis. We find in the course of time that some of these biomarkers are actually measurements of early changes associated with the disease. We'll call them biomarkers of disease. And the others are biomarkers of exposure that are potentially causal. And they can be derived from any of these sources and a variety of others. Again, causal biomarkers versus reactive biomarkers. So I'm at the end. What's the future of the exposome and this world of disease etiology? Well, I think transformative research happens once in a generation. And between 1988 and 2010, there's no doubt that genomic research dominated investigations of disease etiology, and despite disappointing results. There have been almost 2,000 GWAS published in the last 20 years. I think exposomic research via EWAS will find the causes of disease. In fact, some of them already have, and could dominate the next generation of etiologic research. But this is going to require integrated omics technologies that measure chemicals comprehensively and efficiently in appropriate biospecimens, combined with advanced bioinformatics. And it's a big enough idea and a big enough project that we need to get cooperation from government, academia, and industry, especially the industries that are involved with technology development. We need to find some mechanism for establishing workable partnerships for all of these groups to contribute to this vast goal of understanding the causes of chronic disease. So with that, I thank my collaborators, Paulo Vanez, Dinesh Barapal, Augustin Scalbra, David Wishart, who's here, Anthony Mascherone, who's here, and in my lab, and Martin Smith, who's my longtime collaborator. So thank you.